Alexis, to tell if an R chain is hydrophobic or halophilic and polar or nonpolar, you need to look at the R chain. So the first thing to determine an R chain if it's hydrophobic, you need to see if it's C, H, or S. That means it's nonpolar and it's hydrophobic. For the polar side chains, to see if it's hydrophilic, polar side chains are hydrophilic and they will have oxygen. And a charged side chain is hydrophilic. It'll be an acid and base, gain and lose electrons. Hydrolysis, water in, monomer out, condensation, monomer in, water out. Electronegativity goes from oxygen, nitrogen, carbon, and hydrogen are equal. Chapter 3. Let's see. Let's get chapter 4. Chapter 4, DNA and RNA. Oh, you're gonna have to draw nucleotides. We'll learn about that in 2 months. Nucleotides polymerize to form nucleic acids. Condensation of nucleotides monophases. Non-spontaneous reactions require energy. When you combine nucleotides, it's a condensation reaction. A always pairs with T and G to C, and then when it's RNA, it's U to C, I believe. DNA is stabilized by hydrogen pairs. Most prokaryotes have one circular chromosome, and eukaryotes have multiple chromosomes that are linear. What else? How does DNA replicate? Complementary base pairing allows each strand of a DNA double helix to be copied exactly, producing 2 identical daughter molecules. This is RNA, Alexis. RNA, like DNA, has a primary structure consisting of a sugar phosphate backbone. RNA contains U instead of T, and RNA contains ribose instead of deoxyribose. The presence of an OH group in the ribose makes RNA. And RNA, U pairs to A and G pairs to C. RNA can function as a catalytic molecule, and our ribosomes are enzymes like RNAs. Capable of catalyzing a number of chemical reactions. The tertiary structure of a ribosome. DNA, the first form of life, is RNA, not DNA. Alexis, don't remember that. The theory of chemical evolution. Life began as a naked self-replicator, a molecule that existed by itself in solution without being enclosed in a membrane. To copy itself, the first living molecule had to provide a template that could be copied, catalyze polymerization reactions that will link monomers into a copy of that template. RNA is capable of both processes. Now you need to draw a structure of RNA molecule made up of U, A, and G. G is on the 5 end and A is on the 3 end. Indicate bonds formed during condensation reactions, bolder with a different color. You're going to do that in biology too, so stick to that. This is chapter 5 on carbohydrates. Carbohydrates are organic compounds with empirical formulas. They have a carbonyl group and a hydroxyl group. A monosaccharide is one molecule. A disaccharide is 2 monosaccharides. Oligosaccharides are short chains, and polysaccharides are long chains. Aldose is found at the end of a monosaccharide, and ketose is found in the middle of a monosaccharide. So it's going to be C with the double bond oxygen on the top for an aldose, and C double bonded to O in the middle of a ketose. That's how you tell the difference. And then to tell if something is a triose, pentose, or hexose, it'll be the number of carbon atoms present in the molecule. The monosaccharides differ in the arrangement and the number of their hydroxyl groups. So isomers, you have glucose and galactose. Same molecular formula. The C bonded to the hydroxyl group, OH, is up for glucose and it's down for galactose. And then if you have an alpha glucose, they're both down. And if you have a beta glucose, one's up and one's down. Two alpha glucose molecules make one maltose. It's a disaccharide, and it loses water in the middle. Hopefully she'll give you the formula, because that's a lot. But I'll tell you the formula anyway. Just don't let me forget to tell you the formula. And then maltose, it's down. It's an alpha.